Okay, on nervous system part 3, here's your data question. And here's the question. Okay, is anybody not done with this question? If so, we need to pause that video. Okay, the question's asking you why on the 4th was it successful? And if you look at the 4 options, the first sentence is the same on all 4 of them. The first 3 attempts were unsuccessful because each individual dendrite did not receive a strong enough stimulus. So you know the reason it didn't go was because it didn't receive the strong enough stimulus. And that's on all 4 of them. Okay, so why did it go on the 4th one is your question. So somebody tell me which one you could eliminate right off the bat. And you should have been able to eliminate A because it says the stimulus energy received by one of the dendrites was increased. Okay, and then which other one could you eliminate? Good, C is wrong too because of axons. Okay, so when you're looking at this question, you can tell that C is wrong. Summation is not received by the axons, it's received by the dendrites. So when you read your notes last night, you noticed that the summation is where the stimulus was received by the dendrites. Okay, so is it because the 4th was successful because all the dendrites received a stronger one? Or because all the dendrites were able to put it together? Correct answer should be D, because when you read your notes last night, which I know you all did, it talked about when the stimulus is all added together. Okay, before we go too far, take a minute, pause the video, and look back over all these words. You want to know the definition to these words. Y'all discuss those for a second at your table. Okay, let's quickly go over these. It looks like y'all got them. Okay, tell me, Bailey, action potential. I'm ready, go. Tell me action potential. That's where the nerve impulse is actually sent. Do you know what number it is? What numerical value? Action potential? Positive 30. Good. Positive 30. Okay. Tell me resting potential.
It's when the neuron's not doing it.
Yeah, it's when the neuron is not activated, not sending an impulse. What numerical value? Oh, one thing we didn't talk about, I do want you to know, resting potential, what does that look like? What does the neuron look like? Or where are the molecules, maybe I should say? Where are those ions?
Sodium is outside, potassium.
Sodium is outside, potassium is inside. Make sure, I know, do what? Yeah, I know we remember that, salty banana, but if you put salt, that's not going to be right. Because salt is sodium chloride. And salt is just sodium on the outside. So that's just a way to help you remember it. Sodium, like salty banana. But it's not really salt, it's sodium. Y'all got that? Okay, so we got resting potential was negative 70, salty banana, and no nerve impulse being sent. Okay, hyperpolarization, Annika.
That's when you let too much potassium.
Good, too much potassium out. Depolarization, Marissa. Stimulus is going to, is being received. Sodium's going out. No, sodium's going in. I'm sorry. Sodium is flooding in. Okay, repolarization.
Repolarization. That's when potassium gets.
Good. Potassium starts to be pumped out. Oh, how on depolarization and repolarization, how are these ions moving? Through the ion channels. On resting potential, how is that being moved?
It's going in and out through the
Good. Sodium-potassium pump. So you want to know that. Resting potential is sodium-potassium pump, while depolarization, repolarization, those are gated ion channels. Okay, tell me what is threshold? The point of no return. It's got to reach that in order to get the action. Negative 55. Okay, excitatory conduction. the impulse jumping down the nodes of Ranvier, right? Okay, sensory neuron. Okay, sensory neuron. Yeah, that's the neuron that receives. Here, listen to the way I word that so you can get it right. The sensory neuron is the neuron that receives the signal, whether it be a signal of touch or sight or hear or taste or blink or muscle contraction, right? It receives the signal. Okay, interneuron. Good, central nervous system interpreted. Motor neuron.
It's the neuron that takes it from the central nervous system to the motor.
Yeah, it's the neuron that takes it from the central to the effector. Absolutely. Axon. Yeah, it's the one that, she said it's the one that, it's the part of the neuron that sends the message, right? So there's one axon. Dendrite.
It receives the signal.
Good, receives the signal. Probably many of those on a neuron. Schwann cell.
It's like, isn't it made out of,
It makes up the myelin sheath. And what else? What is the function? Yeah, protects it, right? So Schwann cells protects the axon. Okay, we got those words. So take a second and try to answer that question for me. See what you think. All right, what y'all think? C is the correct answer. Good. Any questions on this? Any questions? How do we know that was the answer? Somebody tell me. Good. Effectors had to be last, so that eliminated that one out. And sensory would be how the message is received, right? Okay, if you didn't know anything about this, that's like within, right? That's in between the two other neurons. So inter is going to be in between sensory and motor. Inter, in between. Okay, look at this. Take a second and see if you at your table can say what goes at 1, 2, 3, 4, 5. And not only the word, but like what's actually happening, what ions are moving when. So take a second and pause this. Okay, let's talk about these. Somebody tell me what's going on at 1. Resting potential. You're looking at -70, salty banana, resting potential. Okay, number 2, what's going on? Yeah, it's reaching threshold, right? So I would say sodium starts to come in. The reception of the message is occurring. Okay, what would this, I think this right here is actually showing you threshold. Don't y'all? That's threshold right there. Point of no return, and that's in all animals. Okay, number 3, what's going on? Depolarization. Sodium is where? It's going in. Sodium's going in through the ion channels. Okay, what would this be right here? The question mark. Action potential. Pop goes the weasel. Okay. 4, what would that be? Repolarization. What's happening? What's moving? Potassium's going out. Good. Y'all got this. Okay, what's going on at 5? Hyperpolarization, which means? And then back to rest. Absolutely. Yes, ma'am.
It starts out with potassium inside and the sodium outside, but then it switches and the potassium goes out. How does it get back to that?
Resting potential maintains it.
But it's
Okay, it's all about the charge, getting the charge back to negative 70. So I think the molecules actually are going into the bloodstream, and then the cell reproduces them. Does that make sense? Because looking at this chart, it's all about the charge. It's not about the movement of those molecules. But the movement of the molecules gives it the charge.
So it's just like, okay.
It's just got to get it back to that charge. And those molecules are gone.
You said the salty banana.
I'm with you. No, I agree. Okay, looking at your notes, that was a really good question. Oh, this is the exact same picture Same graph, but it's showing you like what's going on the cell membrane surface. So like 1 right here, if you look at this picture, you should see, what do you see on that picture? You tell me what you see. What do you see on this picture?
You see the stuff going out of it.
You see things going out of it?
You see the sodium's going out. Sodium and potassium are going out. And then, but it looks like...
Somebody go to the board and point to me which sodium is going out. That's what I want to know.
Sodium, it's going in.
Is it? No, it's going in. Where? Look, there's an arrow with sodium going out.
It does? Look at it. It does. What does it go? The arrow, put the little finger on the molecule right there. And it goes down.
Oh, it bounces up.
And it bounces up. So it doesn't get to go.
Okay.
So the channels are closed. Do y'all see that? So look, the molecule goes down and then the channel's closed.
It's just weird.
So it doesn't get to go through. So those are ion channels. There's nothing, the ion channels are closed. The only molecules that are moving through the sodium-potassium pump, which is not pictured. Okay, so 1, resting potential, the channels are closed. Okay, now if you look at 2, so look at this picture. What's happening now? Now Haley says sodium's going out. I see it. Sodium's going in. Sodium's going into the cell. And then on 3, so we're right here on the graph. Look right here. Right there on the graph. Now there's just more sodium coming in. Do y'all see that? Just more sodium coming in. Okay. Look at 4. Y'all told me potassium should be going out, and that's what's happening. But look at 4. Do y'all see that the sodium channels are closed? Yes, do y'all see that? Okay, and then 5 is just showing, I guess, too much sodium came out. So could y'all have done this, looked at that picture, and told what was going on? Okay, look at your notes. Let's go through and look at the things you should already know. First thing, it says diffusion. What is diffusion? Movement of molecules from high to low. It's what kind of transport? Passive transport. Diffusion is passive transport. So maybe write that word in if you didn't know that. Okay, on number 2, it says ligands can cause conformational shape change. What is a ligand? It's a signal molecule, right? Remember, what are the 3 steps to signal transduction? Good, reception. The ligand binds. The G protein has a conformational shape change. Right? That's what the notes are saying. It has that shape change. Transduction, what happens? The message is changed. And then the response is where the cellular response is carried out. So, remember on that last test, I was like, hey, this is going to be on your next 2 tests. Learn it. Reception, transduction, response. Okay, well, here it is. Today's lecture is all about how one neuron communicates with the next neuron. Do y'all remember on Friday when we had those neurons out and I told you to put your nerve impulse at the end of your axon? Okay, that's what we're picking up today. How did the nerve impulse get from one neuron to the next neuron? That's cell-to-cell communication, right? Neuron to neuron, cell-to-cell communication. Well, your last test, the name of the test was cell communication. So neurons communicate is today's lecture. How that happens. Reception, there's a message. Transduction is changed. Response. The next response would be that the next neuron is depolarized. Does that make sense? Yeah, just because you got to get the message all the way down to like wherever. Okay, and then different ligands cause different responses. Yeah, some ligands are going to make a hormone be produced. Some ligands are going to turn on an enzyme or turn off an enzyme or flex a muscle or release calcium or, the ligands do different things. They not only do they have different cellular responses, but they have different receptors also. Okay, so looking at this picture right here, what's that gap called? It's the synapse, right? You know this. That gap is called the synapse. So looking at your notes, it says these are the gaps between neurons. So in order for this neuron, and I'm going to call this one the presynaptic neuron, to communicate with the postsynaptic neuron, they've got to get the message from one neuron to the next. Okay, pay attention to those words I just said. Look, presynaptic is before the synapse. Postsynaptic is after the synapse. Y'all see that? That's easy. You just got to get it. Pre, before. Like if you're in the band, you do pregame. Right? Pregame. But after the game, if when I was in college and we performed after the game, it would be called a post-show. Post-show is the show after the game. Post-show. So, pre-synaptic neuron, post-synaptic neuron. Okay. So, on your notes it says, these are the gaps. There's two types of synapse. The electrical one is all about the charge. But if it has an electrical synapse, the neurons touch. You'll see where it says they touch. It says direct contact. So the two neurons are touching. The chemical one, which is what ours are mainly, are where there's a neurotransmitter and they don't touch. So the neurotransmitter has to jump the synapse or get past the synapse. Okay, it says this requires a neurotransmitter. What is a neurotransmitter? It transmits the signal. What'd you say? It brings the message. It's the message between neurons. And there's actually a bunch of neurotransmitters. There's neurotransmitters that make you happy and sleepy and excited and scared and flex muscles. Like those are all neurotransmitters. Okay, and then it says the signal goes, electrical energy, charge. That's what that means. It goes from charge to, it says chemical energy, which is a molecule. So it goes from charge to molecule back to charge. And that's what we're going to look at today, how it takes the charge and releases the neurotransmitter, and then it goes back to being a charge. The charge is like sodium and potassium ions moving. That's what the charge means. Okay, so if you look at this picture, it's like I can teach all of this lecture with this one picture. Okay, if you look at this picture, remember how on Friday we looked at the nerve impulse going down the axon, and I was like, okay, put your nerve impulse at the end. And that was, what's the end of the axon called? The end of the axon, what's that called? Terminal, good. The end of the axon is called the terminal. So it went down the terminal. So here was like whatever you chose to be your nerve impulse. Remember I was like, just set a cell phone on your neuron. Do y'all remember that? Okay, so when it gets to the end, watch this. Ready? When the nerve impulse gets to the end, calcium floods in. Oh, there it is, going in right here. Calcium goes in. See where it says calcium right there? So calcium floods in. Calcium goes in, and calcium gets the neurotransmitter in these vesicles right there. So the neurotransmitter, like acetylcholine, remember we talked about how acetylcholine was the neurotransmitter flex muscles? So calcium floods in, and the neurotransmitter is in this little bubble right here. What is that bubble called? A vesicle. Do y'all remember when we held hands and we let a food particle inside with a bubble, and then we let it outside? I think it was me and Bailey. What was that called when the bubble came in and the bubble came out? Endocytosis and exocytosis. Great. So that is a vesicle. I called it a bubble, but it's a vesicle. Okay, right there. And this vesicle, look at this right here. This vesicle has in it neurotransmitter, acetylcholine or something like that. Okay, the vesicle, so the bubble, fuses with the cell membrane on the presynaptic terminal. Yeah, look at, we're going to go back over those steps. But look right there where it fuses. See how right here it's fusing? Right there? So the message, let's start from the top. The message gets to the terminal. Calcium comes in, and calcium pushes the vesicle of neurotransmitter to the end of the terminal. Right there. Y'all with me so far? The neurotransmitter goes out into the synapse. Some books call the synapse right here the synaptic cleft. It's just the gap is the cleft. So the neurotransmitter is now in the cleft. Look, you see the little green particles right there? Now in the gap. So the neurotransmitter is now in the gap. Y'all good? Okay, now I'm going to go to another picture right quick. Maybe I am. Okay, look at this picture. So it says the neurotransmitter got to the end. On the calcium is on this side this time. Calcium comes in. There's your vesicle. The vesicle fuses with the membrane. You'll see the vesicle fusing. Neurotransmitter is released in the synapse. So there's the neurotransmitter. Okay, now do you see how the neurotransmitter gets on that little purple protein? Y'all see this little purple protein right here? Okay, look at it over here on this picture. Acetylcholine, or the neurotransmitter, binds to the receptor protein on the postsynaptic terminal. So the postsynaptic neuron. So, yeah, let me say it again. The acetylcholine, right here, this blue dot. Acetylcholine, right here, binds to the receptor protein, and this is on the postsynaptic neuron. So acetylcholine binds to the receptor protein on the, in other words, look at my picture. Acetylcholine binds to the protein on this neuron. And when it binds, look at my picture. What's going on? What happened when it binds? What happened when it bound? Do what? It opens the channel. Look right here at this picture. When acetylcholine binds, here it is, bound, that green dot. When acetylcholine binds to that receptor, it opens the channel. Now look at the picture. What happens? What are them things? Sodium and potassium flood into the next cell. Okay, so from the top, listen to me. Everybody watching? The nerve impulse gets to the end of the terminal. Okay? When it gets to the end, calcium floods in. So calcium's now going to be this pink thing. Calcium floods in. And calcium is going to attach to those vesicles and push it to fuse with the membrane of the presynaptic neuron. And that's going to push the neurotransmitter into the gap. The neurotransmitter goes to the next neuron and binds with the protein. And it's a gated ion channel, just like my magnets for the last test. It's a gated ion channel. When the neurotransmitter binds, the gate opens. And when the gate opens, it allows sodium to flood into the next cell. So now sodium has flooded into the postsynaptic cell. What does that do to the cell? Absolutely, it depolarizes it. So the postsynaptic cell is depolarized. Got it? Everybody understand? Okay, check out this picture. No, not that picture. Okay, so this is showing those green dots at the top. That's the nerve impulse traveling down. Okay, so the nerve impulse traveled down. When the nerve impulse got to the end, what happened? Look at those red channels. What is that red stuff over there? This. Calcium flooding in. Can y'all see the calcium flooding in these channels? Okay, now y'all tell me what this next picture should have on it. What's the next step? It's going to fuse with the membrane and release it into the gap, right? So look, it fuses with the membrane. Right there, do you see it? Oh, no. Right there, do you see it fused with the membrane? Yes? And then what is this in the gap? What was released? Neurotransmitter, right? Like acetylcholine or whatever it may be. And it's going to bind, this little pink dot right here, is going to bind with a receptor protein, open up the gate, and allow what in? Sodium ions. That last picture showed sodium and potassium, but just focus on sodium because you want to know that it depolarizes the second neuron, the postsynaptic neuron. Okay, so reception, cell-to-cell communication. Where was the reception? What was that? Like, if this is cell-to-cell communication, right? Step one to cell-to-cell communication is reception. So what was reception? The neurotransmitter binds. What was transduction?
Opening the protein.
Opening the channel, sodium flooded in. What was the response? The next neuron is depolarized. The response is that now that it's depolarized, what happens now that it's depolarized? The action can go down. The nerve impulse can be sent. That's the whole purpose, right? If you're trying to flex this muscle, you've got to get the neurotransmitter from the brain to relay the message to that muscle to flex. So it's got to travel down several neurons. Okay, check out this video clip. Right here.
Our brains are constantly processing information, which requires communication between billions of nerve cells. Signals are passed from a sending neuron to a receiving neuron at a junction called a synapse. An action potential in the sending neuron travels down the axon until it reaches a synaptic terminal. The narrow gap between the synaptic terminal and the receiving neuron is called the synaptic cleft.
So you know that, synaptic cleft.
The synaptic terminal of a sending neuron contains numerous vesicles filled with neurotransmitters.
So one thing to note, that terminal has the vesicles with neurotransmitter already there. They just got to be sent to the next one.
Chemicals that carry information across the synaptic cleft. When an action potential reaches the synaptic terminal, the vesicles fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft.
So those purple things are neurotransmitters.
The neurotransmitters affect the receiving neuron, changing the distribution of charge across its membrane.
What is that green stuff? Sodium going in. It said, the video said the distribution of charge. But what that is the sodium flooding in, changing the charge of that postsynaptic neuron. Good.
Let's take a closer look. An action potential is propagated down an axon by the opening and closing of sodium and potassium channels.
Okay, oh, no. Whoa. Okay, so that picture was showing you, we can go back to it, don't you worry. Okay, that picture was showing you an axon being depolarized. Wow. Okay, so this is going down the axon. You've got sodium rushing in. I'm thinking that purple is sodium. Sodium's going in, potassium's going out. So this is just the action potential right here.
An action potential is propagated down an axon by the opening and closing of sodium and potassium channels. When an action potential arrives at the synaptic terminal, it causes the opening of calcium channels.
So the green is calcium.
Calcium ions enter the synaptic terminal through the calcium channels. Calcium ions bind to the vesicles containing neurotransmitters.
So the calcium bound to the vesicles.
This causes the vesicles to fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft.
So do y'all see the little purple things being released? That's the neurotransmitter being released into the cleft.
The neurotransmitters diffuse across the synaptic cleft and bind to receptors in the plasma membrane of the receiving neuron. Here, the receptors are ion channels that open. Ions move across the membrane, changing the distribution of charge across the membrane. The neurotransmitters are quickly removed from the synaptic cleft, ending their effect on the receiving neuron.
Okay, so questions on that part? Are there any questions there?
A single neuron can reach the plasma membrane of the sending neuron.
Okay, we're going to do this
changing the
again.
Distribution of charge across its membrane.
But I'm going to be the voice, because I'm better. Okay, so we're going to take a closer look. Okay, so that's the action potential going down the neuron. You see sodium coming in, potassium going out. So sodium Oh, sodium's the purple one. See how sodium's going in and the potassium's going out? That's the red one. Yes? Okay, when the action potential... Did I say it wrong? Red. Oh, okay. So the sodium's going in. So that's red. Okay, I trust you. When it gets to the end, the green floods in. What's the green? Calcium. Good. Oh, man. I'm having a hard time. Okay, so the green, we're going to fast forward to the green again. The green comes in. So when the message gets to the end of the axon, the terminal, the calcium floods in the cell through the calcium channels. And then calcium binds to the vesicles. Calcium binds to the vesicle. And the vesicle fuses with the cell membrane of the presynaptic neuron, releasing neurotransmitter into the cleft. Neurotransmitter then binds to the protein receptors on the postsynaptic neuron, opening the gates so that ions, or sodium, can flood into the next neuron. So this is the next neuron down here. Can flood into the next neuron and depolarize it.
Can we sit in here?
Okay, questions on that? Do you understand? Yes? Okay, now... maybe.
The signals arriving from the
effects of multiple synapses.
A single neuron can receive signals from many sending neurons. The signals arriving from the axon on the left are excitatory. They make the receiving neuron more likely to generate an action potential.
Okay, look at your notes. You see where it says excitatory? That's where it starts. It generates the action. So that's a chemical that just says generate. The very next thing, EPSP. Okay, now listen to this part. Oh, man.
The signals arriving from the axon on the left are excitatory. They make the receiving neuron more likely to generate an action potential, as indicated by the green glows. Signals from the axon on the right are inhibitory.
Okay, so look at inhibitory on your notes.
Less likely to generate an action potential, as indicated by the red glows. The two sets of signals cancel each other out, and no action potential is generated. Now, only excitatory signals are transmitted, and the receiving neuron generates an action potential down its axon.
Okay, so you wanted to notice EPSP excitatory, and then IPSP inhibitory. Stop. So excitatory starts the action, inhibitory stops the action. And then did y'all notice when it went through, like you saw the message was received right here, and went down the axon, and then did y'all see how the message went to that neuron and that neuron? So all of the messages within this neuron will be summated, or the summation of them. So it says summation. This term refers to the adding of dendrite stimuli together to reach threshold. What is threshold?
Point of no return.
The point of no return, the go. The point of no return, that's my term. So make sure that you understand that that means that the action is going to occur. The nerve impulse is going to be relayed. Okay, questions? Y'all got excitatory and inhibitory and summation? Okay.
The signals that pass between these and countless other neurons constitute our thoughts and coordinate our activities.
Okay, look at the next part of the notes. And really, these are just examples of neurotransmitters. So I just wanted you to see them in case you ever were asked a question about them. But before we go there, I put this in here just to make sure you understand it. The neurotransmitter binds, and that's letting sodium in, right? Okay, this says, these are just some words. It says neurotransmitter release is a form of exocytosis. What is exocytosis? Good. Letting molecules, or in this case, neurotransmitter, out of the cell. Does it require energy? Yeah, to make that little bubble and fuse it with the cell membrane, that would require energy. Okay, and then this says the neurotransmitter will then be released from the postsynaptic membrane and degraded. Okay, so that's one we did not discuss on the steps. Look at that 9th step. After the acetylcholine binds right here, if the acetylcholine stayed, then the gate would just stay open. And the sodium would go in and in and in and in and in and in and in. And you don't want that. That would be like a Charlie horse all over. That would hurt. So there's an enzyme. Look at your notes. It says cholinesterase. How do I know that's an enzyme? Hunter. ACE. Good job. So cholinesterase is just the enzyme that breaks down acetylcholine. So that makes acetylcholine go away so that the gate can close. Right?
So how does the next cell get a neurotransmitter?
The next cell already has the neurotransmitters at the terminal.
Okay.
So yeah, did y'all hear what her question was? How does the next cell get the neurotransmitter? It's already at the terminal. It's all like the neurotransmitter is at the terminal of the next cell already. The neuron produced it. Good. Okay, looking at this, this picture is just showing you the sodium entering. See the sodium entering the next cell? Okay, so take a minute. Here's what I want you to do. Take a minute. I'm going to number this. And y'all see if y'all can figure out what's happening at each step. So 1, 2, that one's 3, that one's 4, and that little box is 5. So take a second and discuss those. See if you can figure out what happens at each one. Okay, somebody tell me what happens at 1. Somebody tell me what's going on at 1.
I think, is that where the neurotransmitter goes? Is that the message?
That's where the signal is being sent. The nerve impulse is being sent down the axon. That's what we did Friday in class. We got the nerve impulse to the end of the terminal. Right? Like this all up here had to be depolarized. Okay. Number 2, what happened? Good. Calcium enters. So let's go with yellow now. Calcium enters through the calcium channel. Okay, so somebody tell me what's going on at 3.
The vesicle is formed.
Yeah, the vesicle is formed with neurotransmitter in it, right? Calcium binds to the vesicle. You could say that. Okay, somebody, what's going on at 4? Good. The vesicle fuses with the membrane. You can really see that because I chose orange. Not a good choice. Okay, somebody tell me what's going on at 5. Neurotransmitter binds to the receptor protein, opens the gate, and the next neuron is depolarized or sodium comes in. Do y'all see everything? This is tough stuff. I know it's a lot to learn. Do you see it all? Okay, one thing that's not on here. What happens to acetylcholine now? Sorry.
Bonds with cholinesterase.
It doesn't bind with cholinesterase. Cholinesterase breaks acetylcholine down. So now it's gone. Acetylcholine is broken down by cholinesterase. Good. Okay, try this question. Okay, so somebody tell me, what's preventing the calcium? I mean, when calcium is prevented, what would likely happen? Somebody eliminate one for me first. How about that? Good. A is not the answer because if you prevent calcium, it's not going to increase the neurotransmitter. It's going to decrease the neurotransmitter, right? So the answer is B.
So calcium's never outside of the neuron. Like it's never in the synapse.
Right. No, calcium doesn't make it to the synapse.
Okay, that's what.
Okay, and then it's going to eventually get to the effector cell. So the neurotransmitter is going to get to the effector cell, which is going to make the muscle flex or the gland release a hormone. Okay, looking at this, the last part of the notes, it's all about neurotransmitters. These are just examples for you. You already know acetylcholine. What's it do? Flex muscles, right? Acetylcholine, it says, most common, it can be excitatory or inhibitory. What does that mean?
It can start or stop.
It can flex the muscle or it can release the muscle, right? It can start or stop. Okay, the next one says biogenic amines. What would that be? If you saw the word amines, what would you think? Amino acids, right? So then it says out beside it, these are large amino acid chains. And what's an amino acid? A protein. Good. 4 examples there. You got epinephrine and norepinephrine. Those are adrenaline. Make your heart beat real fast. Did you ever play hide and go seek when you were little? And like you got to your hiding spot and you crouched down? And it was like... It's like your heart was beating so hard and you're breathing and they didn't see you but they could hear you breathing so they caught you.
I thought that was just because you're out of breath.
Well, it could have been because you ran. But it's because you were like so excited and nervous and you didn't want to get caught.
You do.
Do what?
I always have to pee.
Yeah, once you get there you're like, oh man, I gotta go to the bathroom. Yeah, so that's your adrenaline. Like you're laying in your bed, it's dark time, it's nighttime, and thank you, man. You're laying in your bed and you hear a noise and it's like somebody's breaking in the house.
Yes.
But then if that happens at lunchtime, you're like, oh, that was a
I
noise.
thought that was
Do what?
I thought that was one.
You thought somebody was at your house? I think it sometimes too when I hear a noise. So that is, okay, so epinephrine and norepinephrine, those are your adrenaline. It says they work together for fight or flight response. They speed things up. Okay, dopamine, that's your happy. Happy, happy, happy. Serotonin, that's your sleepy. It says ADD and schizophrenia, their serotonin and dopamine are out of balance. Okay, another thing, it says LSD and ecstasy. These are just interesting facts. The drug ecstasy or LSD, it mimics that happy. So the high from ecstasy
Whatever
is...
you're feeling, it just makes it better.
It's like happiness, and you're real sensitive. So, like, I know you probably associated ecstasy with lights. I know we don't do ecstasy ever, but like the glow sticks, and you've probably seen them on movies or something like that. The glow sticks, it's because the sensation is so much faster and neater. So the light is moving around. I've also heard on a different note with ecstasy, like the first time you do it is like the greatest sensation ever, and it feels so wonderful, and the next time it's like you just want that same sensation over, but the second time it's not as good. So it's like you try it a third time and it's not as good as the second time or the first, so you do it again, and it's like that's how people get addicted to it, because they do it again and again and again, because they're always looking for that high, because once you've had that happy feeling, it's like that's all you crave is that happy feeling again, and that's not where you want to be. You don't want to depend on a drug like that. Boo for that. Okay, amino acid. You want to know GABA for sure. That's an inhibitory. It's acetylcholine and GABA are the probably 2 most common ones. Okay, GABA. You want to know that one? That's an inhibitory. Substance P. That's your pain release. Have y'all ever heard or seen the little girl on TV, the girl with no pain? And you think like that's great. You would have no pain. You wouldn't feel anything that hurt.
But if somebody is cutting you and you don't know and you're bleeding.
But it would be bad. Like if you touched a hot stove, you wouldn't know it was hot. And you pick your hand up and all the nerves in your hand would be fried. Or like, you know how when you get something in your eye. I've read the article before. When you get something in your eye, like you know there's something in your eye, so you go to pick it out before it actually scratches your eye lens. Well, the little girl that doesn't feel any pain, she doesn't know that there's something in her eye. So her eyes are like all scratched up and she's blind. Like when sand gets in your eyes and it hurts, you know to go rinse your eyes out because it hurts. But she doesn't feel it. So she would have all scratched up eyes and stuff. Okay, and then endorphins, they block the pain substance. Endorphins are a natural chemical in your body that relieve pain. Natural chemical from the body that relieves pain. So, ready, movie quote. Exercise releases endorphins. Endorphins make you happy. Happy people just don't shoot their husbands. Illegally blonde. You got it. Oh, you got it. Good job. Endorphins make you happy. They release that sense of pain. And then the last 2 are just gases that you can actually take. So when you go to the dentist and they put you on laughing gas, that is nitric oxide. And what it does is it's inhibitory and it prevents the muscle sensations. So it doesn't hurt when you get it relaxes you. So when they're drilling on your teeth, you're not like all tensed up. Now they numb your mouth, so it's not hurting in your mouth, but they give you the nitric oxide to relax you. So you don't like feel everything. But it's like, I'm floating. Am I still touching the chair? You know what I'm talking about?
I had a weird dream when I
Yeah, you went to sleep? I don't think you're supposed to go to sleep.
I don't know, but I was like trapped in a cell in my body.
Inside the
I
cell?
was thinking about I don't know, and I was like, I was in a cell and it was just dark and I couldn't get out.
Oh man, it's crazy. So, but it's like you can't like feel, it's like you can feel the chair up under you, but it's like kind of tingly.
Yeah.
Because those senses are inhibited. Okay, and then it goes away. Don't worry, you're not like permanently floating. Darn it. Yeah, okay, and then carbon monoxide, that's what comes out of your gas exhaust on your car. Do not do this. Do not. Did everybody hear me say that? Do not. Close your garage door. Crank the car. Carbon monoxide will kill you. What happens is an inhibitory, you go to sleep, and the message stops your heart from beating and your lungs from breathing. So you go to sleep. It's not going to happen like if you crank your car, then open the garage. Because my mom always told me not to do that. So sometimes I'd be like, crank the car, I'd be like, oh my gosh, I got to get the garage open. It's not like, it doesn't happen within 0.0 seconds. You have to go to sleep and then it happens. But still not what you want to do. One more thing about the dopamine, the happy. After my father died, my mom and my dad have been married for like 31 years or something. And my mom was having some problems with depression. And so she actually went to the doctor because she had a sinus infection. But while she was at the doctor telling the doctor about her sinus infection, she started crying. Because she was depressed. Like she was just, her chemicals were off balance. And so the doctor told her that he wanted her to start taking he was like, this crazy lady came in for a sinus infection. Now she's crying, telling about her ex-husband. Or not her ex-husband, her deceased husband. And so he told her that he wanted to put her on a antidepressant. Well, she didn't want to take an antidepressant. She just didn't think, she didn't want to use it as a crutch. And you know how you would all feel if you were prescribed an antidepressant. But he explained to her that her body naturally made dopamine, the happy hormone. And her body was making it fine, but that the receptors on her neurons were closed. So in her brain, she was making the happy hormone, dopamine, but the receptors were closed, probably because, and this is just my guess, she was spending so much energy worrying about all the things you would have to worry about after your husband died. Or like she was spending all her energy with sorrow. She wasn't spending energy to open those little receptors, like literally spending ATP. So her receptors were just closed. So she took a medicine that opened up her receptors, and they told her it'd take her about 2 weeks. It wasn't like she was happy the next day. It took about 2 weeks to get her to feeling normal. No, she was still sad because that's normal, but she wasn't like crying all the time. Does that make sense? So that opened up her happy receptors. And now that I teach this, I'm like, oh, I get it. The receptor. You see what I'm saying? Okay, on the back of your data question. Go. On the back